Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Jordan Stillman, Digital Products and Marketing Coordinator for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. Please enter your questions at any time. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Xiao Pan from GenScript Probio.
This is Xiao Pan from GenScript Probio, focusing on process research and development. Today, I'll talk about scalable lentiviral vector manufacturing using proprietary suspension cell line. I'll talk about the following parts. First, I will give you some background on lentiviral vector in the application of gene and cell therapy. Currently, there is a growing number of products with marketing authorization in the recent 6 years. And as can be seen in this table, there are 4 marketed products using lentiviral vector as delivery vehicle. With that, there is currently large lentiviral vector amount needed. Currently, the demand for cell therapies for the treat of cancer is estimated to grow at a high rate from 2020 to 2028 with a CAGR of about 16%. And according to the published data, there is about 70% of the LVV development is focusing on upstream to be able to obtain a sufficient amount of viral particles. Over 40% of the cell therapy programs in preclinical phase, and as these move towards clinical phase, large amount of the LVV is required. Also, there's increasing number of cell therapy programs are targeting solid tumors, and this also will incur large amount of LVV requirements. In upstream production of the lentiviral vectors, compared to the adherent system, the suspension system is characterized by high titer in supernatant, which results in high batch yield. Moreover, there is no residual BSA in the production system, therefore lower the risk of the product. One more thing that is not mentioned here is that the suspension system is more scalable. It's more easier. It's easier to be scaled up. Currently, there are still difficulties in lentiviral vector manufacturing. For instance, the characteristics of the product and intermediate is very sensitive, and therefore the manufacturing process has to be carefully designed to control the process very well, and for instance, to the product loss due to sensitivity and the instability of the product the loss rate is very high. The manufacturing process needs to be carefully designed, and also the analytical method needs to be carefully developed, because for both the functional assay as well as process-related impurities. There are also currently uncertainties in industrial standards with regard to the regulatory requirements. In the next few slides, I will introduce the lentiviral vector production platform in GenScript Probio. Benefit one is we're using a proprietary cell line called Power S293T. This is an serum-free adapted suspension cell line that is demonstrated to be comparable and even better than the top competitor cell line. With regard to the titer greater than 1 to the power of 8 TU per ml and consistent and stable yield over many different projects. As can be seen here, process scale up from 30 ml shake flasks towards 50-liter bioreactors. The cell growth profile in terms of cell density and cell viability are very comparable between batches. Also, the LVV productivity and yield is also they're also very comparable between different scales. Our suspension cell line it results in high crude titer compared to the adherent system, about 10-fold increase, and also the final yield had a 5-fold increase compared to the adherent system. We also tested the T-cell transduction efficiency of our suspension system and compared that with our adherent processes. It can be seen here that the suspension system resulted in high performance in T-cell transduction, and the performance is also comparable with the adherent processes on various projects. In the following slides, I'll give you an introduction of the process development pathway of our suspension lentiviral production system. Before the process development, there are some key points to be considered. The first one is the selection of system, of course. Which cell line do you plan to use? Do you want to use adherent cell culture or suspension cell culture? What's your target amount of product titer? Do you plan to use transient transfection system or stable cell line system, for instance? And there are specific requirements due to your product application or specific quality specifications due to regulatory needs. And in your process design, you have to carefully determine the critical process parameters and critical quality attributes according to your product application and according to your prior knowledge. This will determine the scale-up strategy of the process and how simple or sophisticated of the process is that will impact your process transfer later on. Also, how to evaluate the product, especially the functional assays. In upstream process development, we first look at our cell line performance. It has to demonstrate the good stability of the viral vector production for different, here we demonstrate up to 22 passages stability. Followed by that, we typically do a medium screening for the cell proliferation to support better cell growth. And next, we optimize the packaging parameter that's impacting the LVV production. These are typically cell density at transfection point or transfection reagent used, plasmid ratio, and the ratio between transfection reagent and total DNA. We typically do this through design of experiments with various amount of input and output data through multivariate data analysis. We can interpret the data in a very systematic way. As a result, the upstream process development should demonstrate good stability as well as scalability. Stability is characterized by very narrow bars of CV for different with respective projects with different batch numbers. And the scalability demonstrated here ranging from shake flask to 10-liter bioreactors showed satisfactory crude titer across different scales. In downstream process development, we're focusing on different unit operations. Here, I won't zoom into the details of each unit operation, but we showed here in unit operation, for instance, nuclease treatment, selection of depth filters, including primary and secondary filter, and chromatography step, TFF process, and sterile filter screening. For each unit operation, we determine critical process parameters as well as critical process indicators. And these will guide us towards designing a very robust downstream process. Stable downstream process have to be demonstrated by stable process recovery rate and the stable impurity removal rate. Here we show an overall profile of these two process indicators, and also we showed stepwise recovery rate and removal rate to troubleshoot in case there is deviations or unexpected results. Last but not the least, I will give you an introduction towards GenScript Probio. We belong to a GenScript group. The group is focusing on four business sectors. First is our traditional business service line, life science CRO. And second is industrial synthetic biology products. Third one is our sister company, Legend Biotech, focusing on cell therapy using CAR-T technology. And we are GenScript Probio. We provide biologic CDMO services for both gene and cell therapy section, as well as protein and antibody biologics. In GenScript Probio, we do innovation through collaboration. Our vision is to be your most reliable and sustainable biologics partner. In Probio, GenScript Probio, there are 3 pros summarizes our brand name Probio. We proactively approach our customers, treat our customers professionally, and deliver efficient processes. We have a large number of global partnerships and solid track records. Within 3 years, we've achieved more than 60 plasmid and virus CMC and clinical GMP projects. There are 9 IND approval from NMPA, FDA, and PMDA, over 20 plasmid and virus CMC projects, over 30 clinical plasmid manufacturing batches, and over 10 clinical lentiviral vector manufacturing batches. Notably, we also served over 10 mRNA customers. This slide shows an overview of our plasmid and the viral vector manufacturing facility. In total, we have over 10K square meters facility space. And for plasmid facility, we have 5 to 100 liter stainless steel fermenters and 30 to 300 liter single-use bioreactors. And for viral vector facility, we have 50 to 200 liter suspension production cell line, as well as adherent production cell line. We offer plasmid and LVV one-stop solution, ranging from research and discovery all the way towards commercialization by providing cGMP grade product and processes. As a starting material, we have our LentiHelper plasmids immediately available to you. And basically, the 3 LentiHelper plasmids, they have clear history and traceability. And these 3 helper plasmids are without any IP issue, so they can be very handy to reduce our customers' timeline and to reduce the initial establishing cost. With that, I want to thank you for your attention, and I'd like to take questions if you may have any, please.
Thanks, Xiao. So we have some questions. The first question is, what are the main advantages of the suspension over the adherent culture mode for the lentivirus production?
Oh, okay. First, I think there are much higher density cell densities can be achieved due to the in the suspension culture mode due to more efficient mass transfer, and actually the fact that moving from a 2-dimensional culture to a 3-dimensional culture can help with the scale up of the process. And one more thing is that with suspension culture, you don't have to use serum. That therefore reduce the risk of contamination by adventitious agents and animal components. It also reduces the variability between virus batches because you can use one batch instead of many scale out of adherent culture flasks.
Fantastic. Yeah. Thank you. And we've got another one. Which packaging cell line does Probio use in lentiviral vector production, and how does it perform?
Okay. As shown in the presentation, the packaging cell line that we use is a proprietary cell line called Power S293T, and we use that for suspension lentiviral vector production. Also indicated in the presentation that the productivity and the stability of the lentiviral vector production using this cell line is very good, and it has been demonstrated in many projects.
Fantastic. And what are the main challenges of scaling up lentiviral suspension production processes?
I think going from the upstream to downstream, in upstream process, you have to strictly control your transfection step. One of the things that need to be taken into consideration is that transfection reagent and the plasmid mixing time, for instance, how long do you incubate and what are the incubation conditions. Also the transfection itself, including the mixing and the transfection time, because the transfection does happen in a very short period of time, unlike the very relatively long period of mammalian cell metabolism time. And also the scale down models, you can use some scale down model to simulate the transfection reaction to predict the scale up of the upstream process. For downstream, typically due to high cell density, the high turbidity is a critical issue that we experienced in Probio, and approaches including depth filtration, centrifugation are the steps that should be carefully designed for the removal of high content of process-related impurities is important. Yeah, thanks.
Fantastic, thank you. All right, and I think we have time for just one more question. So what is Probio's plan of further optimizing the lentiviral vector production processes?
Yeah, there are many directions, and we're working on some of them. First is we're working on stable cell line for lentiviral vector production. We believe that the transient transfection is only an interim solution, and when the technology gets matured, stable cell line should be used just like monoclonal antibody production. In up and downstream, we're looking for representative high throughput scale down models for prediction of the large scale bioprocess and more efficient and fast process development pathways. And we're also in downstream process development, we're also looking for more sophisticated purification and concentration technologies. There's also analytical method development and standardization of the quantification and functional assays are something that are critical. We're working on at this moment. Yeah, I think apart from those, we are also working on broadening the characteristics of the viruses for the effective treatment of a wider range of human diseases. Those are basically a summary of what we're working on at this moment.
Well, fantastic. Thanks, Xiao.
Thank you.
And thanks as well to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you will receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.